one of our presence here. I'll be demonstrating a small algorithm which I had developed for the laboratory diagnosis of infectious diseases, and this I used in my microbiology as well as at times in pathology. Like when the patient comes to us with a suspected infectious disease, a laboratory diagnosis requires us to have a sample. So, for laboratory diagnosis of infectious disease, we require a sample. Now, this sample can, in the beginning, be divided into two categories. Sample for demonstration of the organism or its properties. Another would be sample for demonstration of the lesion created by the organism in the body. Okay. Then, for the organism, now we know that for every organism, that organism will have some sort of antigens. It will have some nucleic acids within, and also it can directly be demonstrated. Sometimes it can be grown in culture. So you can have some finite number of steps. How do you go with the diagnosis of this organism? Most common and most essential would be direct demonstration. Demonstrate in this, you can go with either microscopy or you can go for culture. Another would be, if you cannot demonstrate the organism directly, demonstrate one of its antigens. So that can be done. So another would be antigen detection. If still that cannot be done, or we require a more sensitive method, another would be nucleic acid testing. And further, if these things are again very difficult or we require certain some more investigation, we can go for the serology. Another thing which I forgot to mention was, you can demonstrate either the organism or you can demonstrate the property of the organism. Property would include biochemical, effect on animals, and so on and so forth. These can be there. Now, it is very well known to us that for nucleic acid testing there are some 3 methods that can be used all the time. That would be either PCR or DNA probe hybridization, and so on. So I only remember that now. This is how it will proceed. Now for antigen detection, we will have ELISA, IFA, and certain 2-3 methods that are fixed. That is the protocol that we have. Now for lesions, demonstrating lesions in the body, we can divide the samples into 3 main categories. One would be fluid samples, another would be solid tissue samples, and another would be something that has to relate with the whole body. Fluid samples would again, depending on the site of infection, it would be CSF, urine, blood, whatsoever. In solid tissues, we will have biopsy. And means we can have direct demonstration. For example, with Lugol's idea, we had seen certain changes in we can see certain changes in kidney, although it doesn't apply here. But there may be a direct gross change in the organ structure that might be seen. And for whole body, I include this radiology. For example, X-rays and all. So now, when I have to read any infectious disease, for example, if I'm learning, malaria, so the only thing I remember is that the malarial organism, it will have a certain antigen called lactate dehydrogenase, it has certain antigen called histidine-rich protein, and it will have nucleic acids, it will be found in blood. Now by going through all this example, I know that it will usually not be demonstrated, it can be demonstrated in microscopy, it will not be done on culture. Now I notice that there are no specific biochemical analysis that we are doing for it. Antigen detection, since I remember only this organism as a whole. So I can do, okay, these two are the things which have to be taken, so I can go for an antigen detection. Nucleic acid testing, again, it was not very commonly used, so I skipped this step. And serology, not very commonly used. Serology is sometimes used, so that can be done. The lesions in the body, I know it's found in blood, so I'll go for only for blood, and rarely it may go for solids, and this would not be required. Similarly, if there is any other organism, for example diphtheria, so I remember that one of begins with diphtheria and its diphtheria toxin and its Chinese letter pattern. Now only by this small limited information about every organism, just 4 or 5 lines, I'll put it through this entire algorithm every time and I'll get what are the tests that are required. Now this was an example of algorithm which I presented. Thank you.